in the world of obesity, I think both of us agree that fructose and saturated fats are evil and that they are contributing to obesity. And our work suggests this, and I think you've just summarized this, and also to fatty liver. For sure, the data shows, in terms of the practical thing for our diets, that saturated fats can increase fatty liver pretty significantly, probably more than just sugar when sugar is given with a low-fat diet.
Welcome to the Proof Podcast, a space for science-based conversation exploring the health and longevity benefits that come with mastering nutrition, physical exercise, mindfulness, recovery, sleep, and alignment. Facts, nuance, and trustworthy recommendations, minus the hyperbole. Hey friends, great to be here with you. I'm your host, Simon Hill. I'm a qualified physiotherapist and nutritionist with an undergraduate science degree and a master's in the science of human nutrition. Today I sit down with Richard Johnson, PhD, author of Why Nature Wants Us to Be Fat, for part 2 of our conversation about nutrition and metabolic health. If you listen to our first episode, which I highly recommend doing before listening to this one, you'll recall that Rick and I spoke about coming back to further discuss his thesis and cover some other studies that we didn't get time to cover in that exchange. Over the past few months, we've exchanged many an email, throwing study after study back and forth, all of which led to today's conversation, which I must say I thoroughly enjoyed. I find Rick to be a really upbeat person and a passionate medical doctor and scientist, open to discussing his thesis at length, and most importantly, open to having it challenged, something that seems to be becoming rarer and rarer. What is metabolic health? Why is our liver central to metabolic health? What is fatty liver? Is the metabolic health movement overlooking the role of saturated fatty acids in the development of fatty liver and insulin resistance? How can we determine if our liver is healthy? We cover all of this and a whole lot more. As always, all references are included in the show notes. And if you want to watch this, you can do so on YouTube, where full-length videos of each episode of The Proof can be found. Please do enjoy. This is me and Professor Richard Johnson. Thanks for coming back on, Richard. It was a real pleasure having you on a few months ago, and I've enjoyed our back and forths on email since. So glad to be finally doing this.
Yeah, me too, Simon. It's a real delight to be back on your show, and your knowledge is wonderful, and it's been inspiring to have these discussions through the emails.
Yes, no, the feelings are mutual. You're probably getting a little sick and tired of all of my emails, I'm sure. The main purpose of sort of doing this today was really at my end to kind of clarify a few things about our liver and how our diet affects the health and function of our liver and the flow-on effect that this has on our metabolic health and risk of disease. Before we get into the sort of the heart of what you and I have been discussing on email, which really could be summarized best as how do different nutrients differentially affect our liver and metabolic health? I'd like to give you the mic to sort of summarize your thesis for those
Yes.
who maybe missed our last step or perhaps are in need of a bit of a refresher.
Sure. So animals in the wild try to avoid at all costs starvation and dehydration and lack of oxygen. And so these animals, we know very much about these biologic processes when there's no food, water, etc. But what's interesting is it would be much better for an animal to survive if it could prepare in advance for periods of food scarcity. And so our thesis was that there might actually be a biologic process similar to what happens like in starvation or dehydration, but a biologic process that instead tries to prepare you by increasing your fat stores and so forth for a time of scarcity. And what we found was that, when animals are preparing, animals do this in nature, right? So animals will in the fall start gaining weight, especially hibernating animals will dramatically increase their fat stores. They'll become insulin resistant, and they kind of like turn on where they suddenly are eating more, and they kind of like activating a biologic process. And from studying those animals as well as studies in the lab, we realized that it might be initiated by a particular nutrient, and that nutrient is fructose. So fructose is the main sugar in fruit, which we think of as healthy, and I think of fruit as healthy as well. And but if you eat large amounts of fructose, and these animals in the wild will eat massive amounts of fructose, and a fruit that contains this fructose, and that seems to be involved in activating a trigger where they suddenly become hungry. They can't control their appetite. They start eating more than they normally do. They start gaining weight, and they actually block the burning of fat, and they stimulate the synthesis of fat, and they reduce their metabolism so that they can conserve energy better. And they raise their blood pressure a little bit, and actually it's the metabolic syndrome is what happens, and they develop this. And when we block in the animal, when we block fructose metabolism, we can block this switch. And so then, we originally thought it was okay. Where do we get fructose? We get it from sugar and high fructose corn syrup and sweeteners and all these soft drinks. And so we thought that was the primary cause. And certainly there was a strong relationship between sugar intake and the risk for diabetes and obesity and so forth. And then we had the discovery that the body can make fructose as well. So it's not just the fructose we eat, it's the fructose we make. And what we found was that certain foods were really good at stimulating fructose production. And the fructose is produced from glucose. So it's produced from a carbohydrate. So the body makes fructose from glucose. So not surprisingly, high amounts of carbs that are what we call high glycemic carbs can be converted to fructose. And so if you give animals glucose, for example, they will develop metabolic syndrome too. But if you block their ability to make fructose, you can really dampen that. Likewise, we found that salt actually helps stimulate the conversion of glucose to fructose. And, we were wondering why animals like salt licks, and because they're eating kind of poor carb food, but when you give them salt, they can help convert some of their glucose to fructose. And that over time, over a much longer time, can lead to obesity as well. And then we started studying it. We found that that red meats and processed red meats can generate uric acid. And uric acid is part of this pathway that drives the switch, this biologic switch from fructose, because fructose generates uric acid, and certain foods do as well, like especially animal processed red meats. And we found that uric acid could activate this pathway as well. And so basically we discovered that a lot of the foods we like, sweet we like, the salty taste we like, umami, which is the savory taste, is really driving foods that raise uric acid. And so a lot of the tastes we like are to seek foods that actually can have this biologic process that was involved in our, that was involved, is used by animals to gain weight. And then in the process, we also were wondering why, some animals are fairly resistant to fructose, like laboratory animals. You have to give them large amounts of fructose. And we discovered that that humans, have a mutation where we have a higher uric acid. And we found that was responsible for making us more sensitive to fructose. So humans are a lot more sensitive to the effects of sugar. And you can show that. You can make a mouse sensitive to sugar by raising its uric acid. And so we could, we began to realize that we're actually have activated our switch. Vitamin C also sort of blocks this pathway, and it's in fruit. So you say, why is that? Well, vitamin C, when fruit's just ripening, vitamin C, or immature fruit, has high vitamin C, and as it ripens, the vitamin C drops and the sugar content goes up. And And so ripe fruit is much more fattening than fruit that's, tart and so forth, that doesn't have as much sugar. We humans tend to like tartar fruits, which is really good because they tend to be a little bit lower in sugar and higher in vitamin C. So that's good. And there's not much fructose in an individual fruit, and all the good things in fruit that you write about and talk about help neutralize the effects of the fructose. But as the fruit ripens and it really gets ripe, the sugar content goes up, the vitamin C content goes down, and now it can activate the switch, especially if you eat a lot of them. So if you make fruit juice where you make 5 or 6 fruit together, you will probably activate this thing. But if you just eat a single fruit, you get the benefits of all the things in the fruit, the flavonols and the epicatechin and all those things. So anyway, so that's the thesis.
Yes, thank you very much for sharing that. I have a bunch of questions, and I think what we're going to discuss today, at least in part, is some of the human health outcome studies, and perhaps why some of the results in these studies, or most of them, perhaps are different to what you might predict from the animal studies or the mechanisms that you're talking about. And I always do find that interesting because I think the mechanisms are really important. But I'm also aware that, and I'd love to hear your thoughts on this, and this is more just at a high level before we get into specifics. When I think of human studies looking at human health outcomes, for example, feeding people different nutrients, say it's fructose or saturated fat or glucose or polyunsaturated fats, and looking at outcomes like how much liver fat increases by, or what happens to blood glucose, I find that very compelling data because for me, that's sort of looking at the summation of a lot of different mechanisms, many that we might be aware of, many that we're not aware of. And so I put more stock in that type of study. Do you see things differently?
Maybe a little bit, so one of the classic traps in clinical research is if they don't understand the physiology and sometimes the physiology hasn't been discovered at that point, but it's very easy to where you can misinterpret a result because it looks like it's right, but you may not understand the biology. So, for example, there are these, I think you and I talked about this. There are these studies where people will give fructose to people and they will compare it to glucose, like the Stephen Piper study where he looks at comparing the thesis is that fructose drives fatty liver a lot more than glucose. And our work suggests that fructose really drives fatty liver. It's really powerful. Okay. So, but the trouble is that glucose is the wrong control because when you give that, it raises fructose in the body. It's converted to fructose in the body. So you think you're comparing fructose to non-fructose.
Can I just jump in quickly?
Sure.
So I know what you're saying there. You're saying, well, it's not the best comparison because in that study, I think the study you're talking about I think Johnston was the first author. It was a 2013 paper, a high glucose diet versus a high fructose diet. And what you're saying is, well, the glucose gets converted to fructose, so you end up comparing fructose to fructose, or at least at some level.
Yes.
My point, though, here that I want to make is that regardless of what the body does to that glucose, the question we're trying to ask here is there a difference in a real world setting if someone eats a lot of glucose versus eats a lot of fructose? Now, the fact that glucose might get converted into fructose is interesting, but for me, that's somewhat irrelevant to the question here.
Well, let me tell you why it's relevant. So first let's go back to this thing and just to say that there are now studies in people, 2 studies that show that giving glucose to people results in an fairly significant increase in fructose in the blood. and also in the brain. So there are studies that are now showing this. And in fact, we make somewhere between 5 and 20 grams of fructose a day, it seems, based upon these two studies. So anyway, so we are making fructose from glucose. And so if the question you're asking is, does fructose cause fatty liver, or is it any worse than glucose? You're right. If you look at it from that standpoint, you can say, well, both of them cause fatty liver. And I agree they do. High glycemic carbs will cause fatty liver.
Can we just? I want to underline and bold this, because this is a point that I think is lost out there, and I'm listening to a lot of different conversations about fructose online, and many people are saying fructose is very different to glucose, and clearly it's metabolized a little bit differently by the body. But that study that we're speaking about here, they looked at supplying 25% extra energy from either glucose or fructose. And they had, I think it was 2 weeks doing the diet, 6-week washout, and then they did the other diet for 2 weeks. So every person got the opportunity to do both diets. And they did this, interestingly, at both weight maintenance and also at hypercaloric conditions. When it was at weight maintenance, which is really interesting, despite having 25% of energy from glucose or fructose, there was no increase in liver fat, triglycerides, or liver enzymes. But then in the hypercaloric phase, both groups saw increases in all of those, but there was no difference between glucose and fructose. Do you think that people understand this?
Yes, but I also think let's talk about this a little bit more. So first off, you're right. High glycemic carbs and fructose both will increase fatty liver. The high glycemic carbs are working by making fructose. And so, for example, in our animal studies, if we block fructose metabolism with an inhibitor, or like in a knockout, we can block the development of fatty liver even from glucose with an inhibitor of fructose. This is important because several companies, large pharma, are working on making fructokinase inhibitors. My little group's also trying to do that. And what's great about it is they will block the development of diabetes, these fructose inhibitors, from both high glycemic carbs as well as from fructose. So it's fructose that's driving it, but the food could be high glycemic carbs. Now, your question about hypercaloric versus isocaloric is also an interesting question. When we did our studies in animals where we were giving a 40% sugar diet, we could calorically restrict animals. We could actually put them on a diet and they would get fatty liver, like bad fatty liver. And they also became diabetic, compared to animals that were actually getting starch, where we could see that the starch was working, but it was just working slower. So we could see that they were going to get fatty liver eventually. But it turns out that when you eat sugar, it will increase fat stores if you take it out long enough. Now, I don't remember the Johnston paper in terms of the duration, but wasn't it like 8 weeks or something?
It was that was a shorter paper. There are a few that take it out to kind of a month or 6 weeks, but I think that was a relatively short trial.
So it took 4 months in a rat. So that probably would be like a longer time, and maybe not longer. Actually, I would say, but 4 months, was when we saw these changes. And so studies that are short term are always a little bit dangerous.
Can I ask you a question there?
Yes.
So I think it is interesting. So at the moment, based on human outcome data, I haven't seen a single study that has showed at weight maintenance a high fructose or high glucose diet increases liver fat. Are you aware of any studies that have shown that?
So it would have to be so. So the problem is when you give a lot of sugar or fructose, it stimulates appetite, and so if you do it ad lib, energy intake tends to increase. So to do an isocaloric study, it's hard to do it for long term because people, because you're giving a food that makes them hungry.
But there are a few kind of studies that have been able to achieve this or close to this, right? There's, I think, another study, the Parry 2020 paper that I sent you. That was slightly different, but that was looking at overweight folks who were randomized to a high saturated fat diet or a high simple sugar diet, and that was for a month. And then they did a 7-week washout period and switched to the other diet. And in that study, even though there was a little bit of change in body weight, there wasn't much, and they sort of adjusted for that when they looked at where the liver fat, what was causing the liver fat to be increased by, and they saw that the consumption of saturated fat in that at a condition of basically weight maintenance, saturated fat led to liver fat increasing by 39% relative to baseline, whereas the sugar diet. In the sugar diet, the amount of liver fat was virtually unchanged. So it's striking to me that in a short trial you can see saturated fat does increase liver fat, but you don't see sugar.
Well, let me talk about the Parry study. Okay. So the Parry study compared a sugar diet that was 20% fat, 65% carbohydrate with a saturated fat, high fat diet, which was 45% fat and 40% carbohydrate. I'm looking at a sheet of paper here. And in the high fat diet, they actually encouraged fast foods and soft drinks and so forth. And it was also encouraged in both sides. So both sides were actually getting a fair amount of sugar. It's just one group got a lot more sugar. The way sugar works, and this is particularly observed in overweight people, is they develop leptin resistance. So let me just explain the experiment we did to help sort this out, and then it can sort of explain the Parry study. So we did studies in animals, and we found that first off, if I put an animal on sugar, okay, initially it will maintain a normal weight. So if I put an animal on sugar for like the first 2 weeks to a month, what it does is if you give it fructose in the drinking water or glucose in the drinking water or sugar in the drinking water, the calories they get from the sugar water, they will count that, they will reduce their chow intake by the same amount so that their total caloric intake stays the same. They won't gain weight, and it takes 3 or 4 weeks at least for this to work. And if you give the fructose in the diet to a mouse, it takes even longer. But if you put it in the drinking water, it's like 3 or 4 weeks. And then what happens is after about 3 weeks, the animals will suddenly start eating more than they're supposed to. And so at that point, they go into a thing where they're hungry, they're eating more, and they've activated the switch. So the leptin, and we proved that was due to the development of leptin resistance. And so it takes some time before that happens. Now, once they're leptin resistant, they still won't gain that much weight if they're just on fructose. But if you now give them high fat diet, they will get very fat very quickly. Their fatty liver will go up. And that's because they're eating extra calories. And that combination of fat and sugar really helps drive weight gain and fatty liver. But and we've even done studies where we've given the fructose and then stopped the fructose, but at a point where they're now leptin resistant. And then when we put them on a high-fat diet, even without fructose, they will gain a lot of weight with the fructose. With the high-fat diet, because they were previously made leptin resistant. And then it takes like 2 or 3 weeks for that leptin resistance to go away. And what we found is that certain fats, and at least in our studies, like lard, which is more monounsaturated, the lard tends not to cause weight gain on its own until you make them leptin resistant. However, butter fat and saturated fats will cause weight gain independently of fructose. And you can even make an animal gain weight and fat even if they don't metabolize fructose. So it's a very real effect. So saturated fats in particular can cause weight gain on their own. But when we did a study with saturated fat and fructose, what we found is that if we gave fructose, we got pretty good fatty liver. We gave this is over 4 or 5 months. We gave saturated fat, we got mild fatty liver. And if we gave them together, we got, actually, I'm going to say, I have to correct myself. We didn't do that. We gave high-fat diet alone, we got mild fatty liver. High-fat diet plus fructose, we got severe fatty liver. And then if we knocked out fructose metabolism and gave high fat and high sugar, we could block it back to where it was with fat alone. Your point is, I understand your point. Fructose alone, can cause fatty liver pretty well, but it doesn't cause a lot of weight gain in the absence of fat.
Yeah. So this is a really good point for us to kind of, I think, at this juncture to emphasize. I'm definitely not saying that fructose and glucose, simple sugars, aren't a problem when it comes to fatty liver. And also, I think you're talking about weight gain. We've mentioned weight gain a few times, but correct me if I'm wrong here. But what we're really talking about here is the deposition of visceral fat, or mostly visceral.
Right.
So certainly, I appreciate all of that research that you just shared, and I think that's super interesting. Where I have a sort of difficult time reconciling all of this is when I do look at the human health outcome data, and I appreciate you're saying that maybe it's a duration thing to do with how long these trials are set up. But what I see is that there's a bunch of different studies that show quite consistently that high saturated fat diets, even in eukcaloric conditions at energy balance, if you feed someone, a human, a lot of saturated fats, even if they're at energy balance, you can drive up their liver fat, which to my understanding is a key contributor to insulin resistance in the liver, to increased triglycerides or VLDLs, increased blood glucose as a result of that insulin resistance, and a lot of these different sort of metabolic consequences. And whereas with fructose and glucose, these simple sugars, we're only seeing them increase liver fat in a hypercaloric context. And that makes me wonder a couple of things here. How much of fructose and glucose affecting liver fat comes down to calories versus the sort of inherent properties of these sugars, and sort of secondly, why most of the conversation around metabolic health is focused on carbs and low carb diet, where I see very, very little talk about saturated fats.
Okay, so let's just first begin by saying that in the world of obesity, I think both of us agree that fructose and saturated fats are evil, and that they are contributing to obesity. And our work suggests this, and I think you've just summarized this, and also to fatty liver. And when you combine fructose and saturated fat, you get much worse outcomes than if you do fructose with alone or fat alone. And And the, like in studies where high fat is given alone, well, we got to be very careful with the human studies, right? Because there's so much sugar and fructose in the population that a lot of people are already leptin resistant. A lot of people already are eating a lot of fructose. And so when you just do manipulation where you add saturated fat, or, it's not like you're comparing saturated fat to no fructose. They're still eating a lot of fructose. And so it's very hard not to show that it's not still a combination of the two. The only way, Simon, we're going to prove this is like when there's a way to block fructose metabolism. And then what we can do is we can see a saturated fat really still increases the fat level that you think it does, in the absence when fructose metabolism is really blocked. Because like in our studies where we were giving saturated fat, a high fat, high sugar diet, if we blocked fructose metabolism, there was no inflammation in the liver. There was no fibrosis. There was fat, but it wasn't causing, a NASH or cirrhosis or anything like that. So
is your hypothesis there that saturated fat is only a problem for liver fat, hepatic fat, in the context of a background diet that has a high amount of fructose?
I think so. And let's take a look at some examples. People who go on a low carb diet can eat a fair amount of fat. But we don't really, at least I'm not aware. I think there are some cases where they will get fatty liver, and especially if the fat in their diet is high in saturated fat, they might get fatty liver. But certainly a low carb diet is associated, tends to be associated with, not gaining weight, not getting fatty liver, not getting visceral fat. And so there must be something about the carb restriction that's helping this. That's not to say that saturated fat's not a problem. Saturated fat is a huge problem. As you pointed out, it increases apolipoprotein B. It increases LDL cholesterol. It can be pro-inflammatory. It can lead to atherosclerosis. It can raise fat in the liver. So it's not like saturated fat is good. And I definitely in your camp that we should be restricting saturated fat, and animal fats are particularly high in saturated fat. So, I mean, it's a good argument.
I think this is also one of the reasons why, like, the Mediterranean diet done well
Yeah.
is associated with very low risk of non-alcoholic fatty liver disease.
Yes.
Just on that point, though, about ketogenic diets, I think definitely, and this brings us back to like what's better for weight loss, ketogenic diets or high carb diets, and we've got that Diet Fit study we spoke about previously. I would agree with you that ketogenic diets can be good for reducing liver fat. But we have to be careful because this can be confounded by weight loss. A lot of those papers are looking at liver
Right.
fat in the first 6 months. And
Right.
of course, we know that if you lose a significant amount of weight, pretty much no matter what
Yes.
you eat, you're going to reduce your liver fat. But when we sort of pan out and look at more 12 to 24 months, I mean, we start to see people regaining weight on all diets. And when we go a bit further, I think low carb diets in the first 6 months, you do see they have an advantage for weight loss. I would be the first person to put my hand up and say that. But as you sort of go out a bit further, it becomes actually quite a level playing field with other diets, and you'd see adherence drops off. And my issue or my questions there are, okay, as that person is adopting the high saturated fat keto diet and losing weight, great, the liver fat goes down. But we know that they're going to regain weight. They're going to be at weight maintenance or in a hypercaloric situation eventually. How good is this diet that is just, packed with saturated fats from butter and meat,
Yeah.
etc.?
So one of the so first off, some people on a keto diet will eat so much saturated fat that their cholesterol can go up into the 500 range, and there are even cases there were 2 cases way back in the 1930s where these Arctic explorers went on what they thought was an Inuit diet, but it was a much higher fat diet, and their blood became like PMIC, and their cholesterols went to 800. So you can do a keto diet the wrong way, that's for sure. And another thing is that meat proteins, in particular, and especially things like organ meats, and they can raise uric acid. And so just by the purine content, and interestingly, high purine vegetables do not raise uric acid because it's a different type of purine that is concentrated in the plant. So, and if the uric acid goes up, that will activate these pathways, to try to hold on to, to try to activate the switch. And they can't work very well because there's not a lot of glucose to turn to fructose. But as a person on a low carb diet over time, oftentimes they lighten up and they start eating more carbs, and now they can convert those carbs to fructose. And so I think that that there's more than one way to get to the fructose. It isn't just from high glycemic carbs and sugar, but even umami foods can drive fructose production to some extent.
There's another study, Richard, that speaks to your point about the combination of saturated fat and fructose possibly being the kind of double-edged sword, the worst case scenario, I should say. And that was the sober cases. It was a 2010 paper, healthy male subjects, and they looked at 3 groups. 35% of calories from fructose, 30% from saturated fat, or 65% from both fructose and saturated fat. And they were matching the energy between those groups. And these were all hypercaloric, so there was a surplus of energy. And the fructose group had an increase in liver fat by 16%, the saturated fat group by 86%. But then when you combined fructose and saturated fat, it was 133%.
Right. Yeah. So, I believe that when you go on saturated fat, that there's enough fructose in the system to really, there's still probably enough fructose in the system that the saturated fat is just going to drive that fat up in the liver very quickly. And if there's no fructose, it will still drive the fat up, but maybe not as much. So the 86%, what we don't know is how much, fructose were they actually eating, and, was there fructose being produced in the liver? But for sure, the data shows, in terms of the practical thing for our diets, that saturated fats can increase fatty liver pretty significantly, probably more than just sugar, when sugar is given with a low-fat diet.
What's the interaction there? So from a mechanism point of view, if you were going to hypothesize, I appreciate this might not be fully understood, but what is it about, say, fructose sort of interacting with saturated fat to make saturated fat more deleterious in terms of this liver fat deposition?
Okay, so fructose, remember, generates intracellular uric acid, and fatty liver is really linked with intracellular uric acid levels. It's been shown in like hibernating animals and all kinds of things where the hepatic uric acid really is a good marker for how much fat is in the liver. And fructose increases it. Saturated fats increase it a little bit, but the combination really drives it up. Now, interestingly, one of the things that's, is, how does saturated fat raise uric acid, and how do fats do? And there's some data that, ketosis can raise uric acid, but there's also data that when you start getting fat, adipose fat, that kind of creates a local hypoxic state, or a local area with low oxygen, which stimulates uric acid production. And so there is this interesting thing where fat itself may drive uric acid production, and lowering fat can lower uric acid for sure. So there is a relationship of fat with uric acid. I mean, there were studies done many years ago showing that putting people on a high-fat diet can raise their uric acid levels. And so, I was looking to see does saturated fat increase uric acid more than polyunsaturated fat. I couldn't find any data on that, but I do know that, like in our study where we gave saturated fat, we measured hepatic uric acid, and it went way up with the combination. So it could be that. And the way the uric acid works is it causes oxidative stress to the mitochondria, which is the energy factories, and then that actually stimulates fat synthesis and blocks fat degradation. But when you're eating fat, you also can, just the fat itself can end up in the liver just from eating a lot of fat. There's some evidence that might be a mechanism too.
Richard, from a survival point of view, I mean, you mentioned animals, what they do in the wild sort of before going into hibernation is, sort of indulge, I guess, on food and build up these fat stores. Do they preferentially store it as subcutaneous or visceral fat? And I'm sort of wondering here, like what would be the mechanism or the biological rationale for storing fat viscerally from a survival point of view?
That's a good question. It does tend to store it viscerally. Fructose does, and so I'm not exactly sure why. I know that in some animals, subcutaneous fat can act as an insulation, so it can raise body temperature. So, for example, animals in the desert tend not to want to put fat in their subcutaneous, or because it can raise body temperature, because it acts like an insulation system. So they'll put the fat in their tail or in a hump or on their back and so forth. Whales actually try to put it more for insulation. So part of it might relate to the survival, the trait of that particular animal.
Have you come across Roy Taylor's he talks about a personal fat threshold and how some of us may have more capacity to store subcutaneous fat. And if that's you, that's, I guess, you're lucky. Whereas others at a same level of body fat will store it more preferentially as visceral fat. Is that something that you think exists?
Yeah, well, there's these studies that show that glucose tends to if it's not converted to fructose, glucose tends to drive fat in the subcutaneous tissues, whereas fructose tends to drive it in the visceral tissues. And that's that beautiful study, that was done by Kim Stanhope in people. And my belief is that if there are people who tend to put it subcutaneously, they probably have better vascular and endothelial function where they're resistant to the effects of fructose. So you can have a fair amount of fructose on board, but not have it activate your switch if your mitochondria are really healthy. So like when you're younger, sugar doesn't do as much to you because your mitochondria are very healthy, your vascular function is very healthy. And even if it does induce a little bit of oxidative stress, it's not enough to activate this switch. But what we currently eating a lot of fructose over time, then it starts to build up. So there are definitely people who are resistant to fructose. So if you see a study being done in a 20-year-old who's lean, and there's lots of studies that are done by this, it's kind of a favorite tactic of the sugar industry, is you pick someone who's got beautiful vascular function, and it's really pretty hard to show an effect of fructose in the short term in these people. If you're like a super athlete, you can drink soft drinks pretty easily without any evidence that it's doing anything because your mitochondria are so super. But if you give this repeatedly over time, it starts to wear down those mitochondria. They start getting, the oxidative stress results in a more profound inhibition of mitochondrial function and so forth. So there is this kind of transition. The other thing is that over a long period of time, the mitochondria will actually can decrease in number. And so you can kind of get locked in. And then the effects of fructose may not be so obvious because you're already in a low mitochondrial state. So, for example, if you've been obese for a very long time, you may or may not be able to show a big effect of fructose because your mitochondria are already, there's already very low intracellular ATP levels. So actually, we did a study in diabetics, and you can kind of see the reserve of how it happens, because uric acid, if you give fructose and uric acid levels go up, that means that you're depleting the ATP and activating the switch. But the trouble is a lot of people, once they become diabetic, their ATP levels are already very low. So it's very hard to actually see that rise in uric acid. And so the rise in uric acid with fructose disappears as your body gets, as your mitochondria go down. The good news is, as you know, you can rejuvenate mitochondria. And that is absolutely the way to go for people with, been obese a long time. So things like intermittent fasting or low fructose diet, low salt diet, those all can stimulate mitochondria. Vitamin C does. But your best thing is exercise, and exercise really helps boost those mitochondria.
Yeah, I've been diving deep into the exercise with biochemists and exercise physiologists, and talking about zone 2 training for improving the mitochondrial efficiency, and zone 5 training, or HIIT, that high intensity training for stimulating mitochondrial biogenesis, so getting more of the mitochondria. So this is a nice kind of overlap here with what you're talking about. I want to come back to kind of zoom out and sort of make sense of this from a very practical level for the listeners. But something else that I do want to touch on is the type of fat, because you've mentioned a couple of times a high-fat diet has done this, and there does seem to be this kind of anti-polyunsaturated fat type rhetoric online, and I can't personally make a lot of sense of it. And when I was looking through a lot of these trials with you, I was finding some pretty fascinating results. And there's a couple studies here I'd like to go through, because it seems that when it comes to hepatic fat, that the type of fat matters a lot. And there was a study, and I'd like to discuss with you, and you sort of spoke about uric acid before, but if there's potentially other mechanisms here at play. The Bajermo 2012 paper, I'm not sure if I pronounced that correctly, but BJERMO, it was a 10-week study, 67 odd participants that were abdominally obese adults. And really neat, they randomized them to a high saturated fat or a high polyunsaturated fat diet. And that polyunsaturated fat diet was mostly omega-6 fats, the ones that tend to get demonized the most. And the goal was weight maintenance, but they did put on a little bit of weight. It was under a kilogram, but over the 10 weeks. And in the saturated fat group, liver fat went up by 7% relative to baseline. And in the polyunsaturated fat group, it went down by 9% relative to baseline. And these researchers were also interested in looking at inflammation and oxidative stress. And so online, you'll hear quite regularly that omega-6s are bad when it comes to inflammation and oxidative stress, but they found no increase in either of these in the polyunsaturated fat group. And their conclusion, which I have here, was that compared with saturated fat intake, omega-6 polyunsaturated fats reduce liver fat and modestly improve metabolic status without weight loss. A high omega-6 polyunsaturated fat intake does not cause any signs of inflammation or oxidative stress.
Yes, I agree with you. So let's just go back. I believe fructose is really playing a big role in driving metabolic syndrome, but it works best when it's given with fat to make works best, meaning creates the most fat when it's working with a high fat diet. But the type of fat really makes a difference. And when it comes to calories, fat is all 9 calories or so per gram. But when it comes to the type of fat, it really does seem to make a difference. And saturated fat really seems to be the worst culprit. And when you compare it to polyunsaturated fats, the saturated fat diets that when I've seen this, like the study you talked about, which is a great one, the saturated fats tend to cause more a worse fatty liver, evidence of more inflammation, a greater rise in LDL cholesterol, all the bad things. So saturated fat tends to be worse. And you're right that, there's some data that omega-3 fatty acids can really, like help block fructose effects. And there are all these studies. I've never done one of these studies, but there are lots of studies suggesting that omega-3 is really good. And that led to this concept that the omega-3, omega-6 ratio is really, really important. And these days, a lot of people are eating seed oils that are rich in omega-6. And so there's all this stuff in the internet saying that the omega-6 is really a big problem. But if you actually dive into the literature, there's not so much evidence that omega-6 is doing anything, bad compared to, other fats. Like saturated fat looks like it's the winner for being the worst loser. It's saturated fat appears to be the much worse than polyunsaturated fats, and monounsaturated fats like olive oil seem to be pretty healthy. So my guess is that that polyunsaturated fats are getting a bad shake, and that omega-3s clearly look beneficial. Omega-6, most studies suggest that they're not as bad as people are saying. But saturated fats, when you take a diet high in saturated fats, you're going to get into trouble. It's going to cause fatty liver. It's going to, it's going to raise your LDL. It's going to be associated with atherosclerosis and increased risk for heart disease. So we do need to restrict our saturated fat.
Yeah, I think, beautifully summarized. I think that where some of the confusion comes, and I can sort of understand how someone could be confused by this, is that there's a difference between an omega-6 fat sort of on its own and the effect it has on health versus where a lot of omega-6s are found, which is in ultra-processed foods, which is a whole food matrix. And we know that ultra-processed foods are deleterious for health, but there are many things in those foods other than the seed oil, which does show up on the label, but they lack fiber, they lack protein, they lack water, they have often added glucose and fructose. So,
and salt.
So there are many reasons why they're probably hyperpalatable and leading to the poor health outcomes, or associated with the poor health outcomes that we see. This is great, Richard. You've clarified a lot of things for me. I want to kind of finish here on metabolic health and sort of tying all of this together, because I realize that if someone sort of jumped into this and perhaps didn't listen to our last episode, we've gone straight into the deep end here with a lot of these studies, and we're talking about liver fat, and blood glucose and triglycerides, etc. Can you just define to me, what does metabolic health mean to you? If someone that doesn't have metabolic health has metabolic syndrome, like, what is metabolic health?
Okay, well, to me, metabolic health is when your energy factories that are producing ATP are working at a very high level, and your intracellular energy levels ATP levels are good. So I think metabolic health is really an energy condition. So what fructose does is, think of energy as stored energy and active energy. And active energy is our ATP, and that's what makes us do everything we want to do. And stored energy is there to help replace the active energy as it's being used, if there's no food around in particular. And what happens is in normal health, ATP levels are high in all the cells and they remain high. So what happens is when you take in calories, they're used to keep that ATP level at a high level. And, there's metabolic flexibility where the stored energy in the form of fat and glycogen, they can replace the energy as needed, and it can go back and forth with ease. And so the ATP levels are high. When you eat fructose, it tricks the cell into reducing the ATP levels. And we've showed exactly how that happens. And once the ATP levels are low, what happens is it sets off an alarm that says, hey, I want to gain weight. I want to store fat. I want to become insulin resistant. I want to raise my blood pressure. I'm going to do all these survival things because my energy levels are dropping and they're sensing the ATP. And when that happens, what the fructose does that, it actually, when you eat the food, instead of replacing the ATP, it shunts it into fat and only replaces a tiny bit of the ATP so that your ATP levels stay low and you keep eating until finally the ATP levels get back up to normal. And then but at the expense that you've now eaten more and you've eaten a lot more than you normally would. And so then you eat more fructose, the ATP levels come down, and then you get hungry and eventually you kind of get locked into that. So to me, it's an energy disorder. And what's interesting is studies of people with diabetes, studies of people with obesity, studies of people with fatty liver. If you measure the liver ATP levels, they are low in all conditions. And that's what fructose does. And that's why fructose is initiating this. It's not the driver of obesity in the sense of clinically. The fructose sets the stage. It creates the metabolic unhealth where now when you eat fat, and especially saturated fat, that drives up the weight gain, the fatty liver, and all those things because you set the system to a low energy level. And actually it's only a low ATP level. The total energy, by energy balance is the active energy plus the stored energy. So diabetes is actually a high energy state, but just that it's all in fat and you can't release it to restore the ATP levels. So to me, I think that there will be a day when people will want to have their intracellular ATP measured, because that is probably the most important measurement for metabolic health. And, if you're a super athlete with great mitochondria, you have scintillating ATP levels. And, and if you're been diabetic for a very long time, your intracellular ATP levels tend to be low. And the liver is the heart of it. When we did our studies, if you knock out the fructose metabolism in the liver, the ATP levels come up in the liver, and it's associated with not just metabolic health for the liver, but reduced. It actually, the way fructose is working, you drop the ATP levels in the liver, that can drive the metabolic syndrome through the whole body. So the liver is sort of the heart where it all begins, when it comes to energy balance. So maintaining good ATP levels in the liver is really the key.
Quick question on future studies. So let's say there's a study that comes out, because from what I'm hearing, you're saying background fructose intake is really the problem here. It's causing this reduction in ATP, and I'm curious, would you have to change your hypothesis or the way you're looking at this if you saw a study on healthy adults who had a low fructose background diet that were fed high saturated fat and their liver fat increased substantially?
Yeah. It seems like you're being a little sly here.
I'm not trying to sneak in a study. I'm just curious, as opposed to if in the future a study like that did come out, would that would be a little sly if somehow I pulled out a study that was published last week. I can assure you I'm not doing that. All these studies we've exchanged on email and spoken about. So
yes.
there's none of that.
Well, if you're so I think that saturated fats can increase fatty liver in the absence of fructose. I really believe that. We did a study with a fructose knockout mouse where it can't metabolize fructose, and if I give it butter fat, it will get fatty liver. Not much inflammation, but it will get fatty liver. And so I do believe that fats can drive fatty liver. I don't know if they drop the ATP level. And and as we mentioned, fat could potentially increase uric acid, which actually will help drive the ATP down. So it could still be a low ATP state with just fat alone. So it may be that fructose is not the only thing that can activate the switch. It's possible that saturated fat may have some effect as well. The fact that saturated fat alone can cause some insulin resistance would be supportive of that. And evolutionarily, eating saturated fat might be a good way to increase your fat stores. So, it might engage some of these same pathways, and perhaps it's through its ability to raise uric acid. So, I think that we have to be open, and my work and the work of my collaborators is based on our, interpretation of the data that we've done, and we're open to modify the hypothesis for sure. But, it does look to me like all these metabolic diseases are really energy disorders where we're dropping the energy in the cell. And if we can stimulate the energy ATP levels to go up, we can really make a difference. And this is why exercise is so good. This is why, reducing the bad foods will help. And it isn't just sugar, it's high glycemic carbs. It's really salty foods. It's, alcohol will do it. And and some of these umami foods like liver, organ meats, processed red meats. And, so there's a lot of things that can do it.
So you mentioned there that the liver is sort of central to this kind of metabolic health puzzle that we're talking about. I'm not sure that everyone fully appreciates the role of the liver. How would you sort of go about summarizing in lay terms, if someone said to you at dinner, Rick, tell me about, how is the liver involved in sort of the management of glucose and fats, or glucose and lipid metabolism?
Well, the liver is the first main organ that when we eat food, the nutrients go into the liver. And this is the place where a lot of the processing of protein and fats is occurring. And so the liver has really been always recognized as the metabolic organ. And it's the main organ that creates ketones, and it does a lot of the gluconeogenesis that produce glucose. It's really involved in many metabolic pathways. And when we give fructose, we see reductions of ATP, not just in the liver, but we see it in the brain, and we see it in the kidney, and we see it in the vasculature. So for sure, the fructose pathway is working through all of these different places, and probably is important in all those different places. But the reason we think that the liver is the king is because we did a study where we knocked out fructose metabolism in the intestine, the brain, and the liver. And so we had animals that could metabolize fructose everywhere except the liver, for example. And when we did that, we found that we could block not just fatty liver. If we gave them fructose, the ones that could not metabolize fructose in their liver were protected from fatty liver. They were protected from insulin resistance. And then, interestingly, they were protected from weight gain, and they were protected from fat accumulation in their adipose tissue. So it was kind of a strange finding where just blocking it in the liver could affect weight and adipose tissue, and it kept the animals leptin sensitive. And when we studied the leptin resistance, which is what drives people to eat more, it's due to a problem in the hypothalamus of the brain where they get inflammation up there. But if you knock out the fructose metabolism in the liver, they don't get the inflammation in the brain. So there's some kind of liver-brain communication that's involved in how fructose works. And if you block fructose metabolism in the liver, you get benefits above and beyond the liver. You end up regulating your food intake, and maybe that's has a role in preventing fat and weight gain. And you prevent insulin resistance from developing. And so, yeah, I think the liver is really a major site for the metabolic syndrome. And it may not be the only site. We know that there are things going on in other tissues, but it's like the king.
Right. You mentioned insulin resistance there. Is it the accumulation of liver fat or hepatic fat what that's primarily driving insulin resistance in the liver?
I don't really know because they tend to go together. And the fact that saturated fats can increase liver fat and cause mild insulin resistance, is another thing that kind of links the two. It's very hard to separate it. There's evidence that it's linked with oxidative stress to the mitochondria in both situations. So there may be like a common pathway till the very end, Mm-hmm. So where they separate. So I think that we should view the two as being intricately linked.
Yeah. I guess when I think of the liver and its involvement in sort of glucose homeostasis, I often think of an analogy to do with the bathtub. I'll share it with you and you can tell me what you think of it. And you may have heard this before. Sort of, you can imagine a bathtub and the tap is the liver. And when it's running, that's sort of glucose going into the blood, which into the sort of water building up in the bathtub. That's the blood, that's the glucose in the bloodstream. And the drain at the bottom of the bathtub is muscle. It could be other cells. But so glucose can sort of build up in that bathtub, which in this analogy is water, if either the tap is unable to be turned off, which is at the side of the liver, or the drain cannot be opened, which is at the side of the muscle.
That's great. Yeah, it's absolutely true. We were doing some studies, and clearly in diabetes, the uric acid seems to have a role in driving the gluconeogenesis, the production and release of glucose from the liver is being driven a little bit by the uric acid. But there's also another player. And the third player is that in metabolic syndrome, not only is there, you've got this resistance, the muscle's not taking up the glucose and there's more coming out from the liver, but also the insulin release from the pancreas starts off to compensate, goes up and up to try to compensate. But over time, the islets in the pancreas that make insulin get weathered down, and there's oxidative stress going on in the islets, and there's a loss of the insulin secretion, and there's some scarring in there. And we've shown that fructose can do that. So if we fed in that study where we gave sugar to rats that was on a caloric restriction, we could show that there was actually the high sugar fed animals were developing islet dysfunction. But again, remember that was a 4-month study in a rat. So that's, it would be like a long study in a human, and that hasn't been done.
What's the problem with high blood sugar? You mentioned blood sugar then, or blood glucose. Why is this such a problem for human physiology?
Well, I think that the problem isn't so much the high blood glucose, but the fact that there's the uptake of the glucose into the skeletal muscle is being blocked, and basically that the problem is the intracellular metabolism. Again, that the ATP levels are low, and that the nutrients aren't getting in there. Mm-hmm. But it's more than just the glucose. It's really the intracellular machinery. One of the things that's really interesting is in the brain, some of the brain is responsive to glucose alone, and some requires insulin to move the glucose into the brain cells. So certain regions of the brain are insulin dependent, and other areas are not. And when you become insulin resistant with fructose, you're also becoming insulin resistant in the brain for those areas of the brain that require insulin. And so certain areas of the brain are actually, the metabolism is lower because there's not enough insulin action to bring the glucose into the brain cells. So glucose levels can be high, but they're not actually getting into the neurons. And it's been shown, it's very interesting, Simon, but the areas of the brain that are insulin dependent are involved in self-control and, a lot of these things that are involved in kind of countering the foraging response. So when you're foraging, you actually want to go into new areas. You have to go into areas you've not been before. So there's exploratory behavior. You have to look around a lot. You can't deliberate. You have to be able to overcome, the recognition that it could be a dangerous area. So you don't want a lot of self-control and willpower. You want to lessen that so that you're more likely to go in to get that food and risk finding a predator. You may have to be a little more aggressive. And so the areas of the brain that are inhibited by fructose are the areas that are involved in the self-control, recent memory as well, where you don't really want to vividly remember what was in that area that you're going to go into now. And so whereas the areas of the brain that are involved in foraging, like there's this part of the brain called the anterior cingulate, fructose doesn't inhibit, the activity there because it wants a foraging behavior to be maintained. What's really interesting is glucose has the opposite effects. So glucose actually suppresses the foraging response, whereas fructose activates it. And what's interesting is that, if you reduce the energy metabolism to these areas of the brain, and you do that chronically, you can start seeing mitochondrial problems, neuronal loss. And those are the exact regions of the brain where Alzheimer's occurs. And the areas that are involved in foraging, like the anterior cingulate, are spared in Alzheimer's. And so recent studies have shown that fructose levels are high in the brain of Alzheimer's patients, especially early in the course when there's still metabolism going on. And so my belief is that the high glucose in the blood isn't really the problem. It's really reflecting the insulin resistance. And that's the problem. And you're not getting the nutrients that you need to your skeletal muscle, but you're also not getting it to your brain. Mm. So it would be ideal to not be insulin resistant.
Do you think that's why there's some, emerging research looking at the utility of a ketogenic diet for folks with neurodegenerative disease?
Yeah, I think that is probably one way it benefits, is that it's helping to decrease the fructose levels in the brain. And like things like a continuous glucose monitor and things like that, where you can kind of figure out what foods trigger your glucose level to go way up. Personally, I think that, if we can keep our glucose levels down, we can reduce the amount of fructose we're producing. As I mentioned earlier on, that there's a study done by Dr. Sherwin at Yale where he took people and he raised their blood glucose, clamped it at like 200 milligrams per deciliter, and showed that it resulted in a marked increase in fructose production in the brain. And we do not want to have a lot of fructose in the brain because all the data suggests that it can, cause memory problems. That you give fructose to animals, they have trouble navigating a maze, and so and it's probably was supposed to help us with our foraging response. So it was meant to be a survival response. And, in the short term, it probably does, benefit. But when you're eating sugar all the time or making sugar all the time, I think that this is what's causing a lot of our problems. So, getting back, I agree with you that saturated fat is a big problem. And I agree with you that polyunsaturated fats, have gotten a bad call, that the evidence is not so great that they're bad. Omega-3 for sure seems to be good, and omega-6, there isn't that much data that it's bad. I agree with you. But I do think that this fructose pathway is a lot more important than we think. And yes, if you just give fructose to an animal, they don't gain weight so much. They, won't get so much fatty liver, perhaps, at least in the short term. Saturated fat is a stronger way to increase fat in the liver in the short term. But all of us are on these diets that have too much fructose. Most of us are either eating or making too much fructose. And if we could just reduce our glycemic carbs, and we could, cut back on salt, drink a lot more water, really reduce our sugar intake, not necessarily eliminate it, of course, but just really cut it back. Plant-based diets are great. I think we could have a big impact. And so the problem with fructose is it isn't just the fructose we eat, it's the fructose we make. So just keep that in mind when we look at these studies.
I think that's a great point. I think it's a telling point. I think if people were to kind of not have read your book or listened to you in detail, they might just think, well, Dr. Johnson's he's against fructose in food, but what you're saying is fructose in general, and that can be through the food or what your body makes. And going back to those studies we spoke about earlier where they compared glucose to fructose and didn't see any difference, you explained that the body will create fructose from glucose, which I think helps clarify that. One thing that we haven't spoken about here, we've spent a lot of time thinking about liver fat, and we've spoken about saturated fats and fructose and glucose and polyunsaturated fats. And I think we agree on pretty much all of that. One thing that we haven't spoken about here, which is part of metabolic syndrome, is triglycerides. And certainly when you do look at these studies where there is a difference, a real difference, or another difference that we haven't yet touched on, is particularly when you look at these diets with excess sugar. In a diet that's providing excess calories, and there's a lot of calories coming from sugar, it does seem to result in a conversion of these excess carbohydrates into fat, and you get this increase in triglycerides. Can you kind of walk us through what's happening here in the liver, and what's if someone's thinking, Doc, what's the problem with high triglycerides?
Well, I'm not an expert on high triglycerides, but it has been linked with cardiovascular risk. It may be hard to separate it from some of the other things coupled with high triglycerides. So high triglycerides is linked with insulin resistance and all the features of metabolic syndrome. And high triglycerides can be associated with conditions like acute pancreatitis and all kinds of problems. So, none of us want to have high triglycerides, and it is viewed as a cardiovascular risk factor, a lesser one than LDL cholesterol, but nevertheless, it's generally viewed as one. And so we try to reduce the triglycerides not only for that, but also to reduce the risk for pancreatitis and things like that. I actually, for a long time, Simon, I was convinced that the fructose pathway was only indirectly linked with heart disease, and that the way it was maybe the triglycerides did it a little bit, but really what fructose was doing was causing it's linked with what we call arteriolosclerosis, raising blood pressure and causing narrowing of the small arterioles, and not with atherosclerosis, where you get these big fatty plaques and so forth. So I always thought that the relationship was more with what we call arteriolosclerosis than with atherosclerosis. But what's happened is I've become more and more aware that the metabolic syndrome and this fructose pathway is associated with a high uric acid. And what's happened is there's been the discovery recently, so high uric acid, is the cause of gout. And gout is this disease that affects like 5% of the population, 4 or 5%. And it's this very bad inflammatory arthritis where uric acid crystallizes into the joints and causes this terrible pain. Gout is associated with metabolic syndrome, and gout can be precipitated by fructose and also high purine foods. And so so I always thought that that uric acid, the crystal problem with uric acid was restricted to the joints and maybe a little bit in the kidneys. But what's happened is in the last 5 years, they've discovered that people with high uric acid levels, and especially with gout, can deposit crystals in their blood vessels. And there's now 2 or 3 studies showing that 85% of people with gout can have crystals in their blood vessels, and they use a special scanner called the dual energy CT scan or DEX scan. And this DEX scan can identify the uric acid crystals. And what's incredible, Simon, is they localize to the atherosclerotic plaque. So if you find it in the coronary arteries, it's usually where there's a cholesterol plaque. And the same thing in the blood vessels, like the aorta. And they've actually done studies and autopsy studies, and the crystals are present in these plaques. And just as cholesterol crystals are thought to initiate inflammation in these plaques, and so your saturated fat diets cause the cholesterol and oxidized LDL, and that's involved in stimulating inflammation in these plaques, the uric crystals may also be doing the same thing. So all roads lead to Rome, and it looks to me like that this could be a mechanism linking the fructose pathway and the metabolic syndrome pathway to heart disease, and not necessarily the triglycerides. They might be, a sort of a marker. They're probably involved to some extent, but it's part of the syndrome. And we now know that people who have a high uric acid in the absence of gout, that about 15% of them have crystals in their blood vessels. And so this also raises the problem of, what happens to the people on the keto diets whose uric acids shoot up? Could they be getting crystal deposition in their blood vessels? And we need to do studies to look at this, because, like on keto diets, you can get, gout attacks are not that uncommon.
Do we know what a healthy uric acid level is yet, if someone was to go and test it?
Yeah, I think we do. So first, it's the intracellular uric acid that really makes the difference. And for example, if you have kidney disease and you're retaining uric acid, the high uric acid may not be the same as if you're making the uric acid. So uric acid, we make it and then we excrete it. And so if your kidney function's not so good, you can retain the uric acid. And that may not be as dangerous as intracellular uric acid, because when it's produced inside the cell, that's involved in the activation of this switch. But if it's elevated just from retention, its main risk may be more of crystallization. But when you have so it sort of depends on what's driving up the uric acid. But in general, if your uric acid is greater than 7 or milligrams per deciliter, which is about 420 micromolar, like in Australia, That's associated with significant increased risk for obesity, diabetes, fatty liver, hypertension. It increases the risk for dementia. It increases, the risk for many, many things. But
If you go and measure that at a standard lab, is that an
yep.
intracellular test, or do you have to ask?
No, it's measuring the serum, but the serum levels are very predictive. But when you do clinical studies where you lower uric acid, you tend to improve things like blood pressure a little bit, insulin resistance, even weight gain can be, like studies in adolescents show that it can reduce weight gain to some extent. So it's probably involved in this pathway. It's not like the cure-all, though. If you lower the uric acid, it isn't that you're going to get a huge benefit, but you seem to get a reasonable benefit, particularly early in the disease. And remember, we talked about how early in the metabolic syndrome, the mitochondria are reversible, and so that if you block this pathway, the mitochondrial function can likely improve. Whereas if you've lost a lot of mitochondria, the benefit of lowering uric acid may not be as clear. So, for example, when we did studies in animals, we found that if we raised uric acid, blood pressure went up. But if we waited a long time, the blood pressure would stay up because of injury that was occurring in the kidney, so that then lowering uric acid wasn't beneficial. It was only in early hypertension. And so we did a study in adolescents where we took 14, 15-year-old kids who were diagnosed with high blood pressure. They were all overweight. And we randomized them to a uric acid lowering drug or a placebo. And we corrected blood pressure in about 90% of them just by lowering uric acid without any antihypertensive therapy. And it was 3% in the placebo group. So it was very, very significant. But that we caught them early when theoretically their mitochondria are still relatively healthy.
Yeah, that's super interesting. As is your theory about triglycerides. I always thought, and actually think what you sort of put forward might help explain things a little more at my end. I always thought that triglycerides were raising sort of risk of cardiovascular disease simply by increasing the number of ApoB containing lipoproteins, because they have to be carried in these VLDL sort of
that could be
lipoproteins that chaperone them in the blood, and that those ApoB lipoproteins are sort of susceptible to entering the intima and becoming retained. But I have seen some studies where they've looked at triglycerides and sort of adjusted for ApoB, and it seems to account for a fair bit of the risk, but there's always a residual risk where triglycerides are still increasing disease to some extent beyond the ApoB sort of element. So that's really interesting.
Yeah, and your point about the ApoB is really right. Seems to be right on too. I like that.
So I want to finish here. Just if someone was to go and do a bit of an audit on their liver, and we've spoken about liver fat. I think in the literature, there's this sort of threshold where people talk about 5% of liver cells containing abnormal amounts of fat, often leading to a sort of diagnosis of fatty liver. So I'm interested in hearing from you on, is there a tipping point where you have too much liver fat, and does someone need to go and do a sort of liver ultrasound, or can they get enough idea by looking at things like fasting glucose and HbA1c, triglycerides, etc.?
Well, there are these. First off, you can kind of assess the level of fat by ultrasound, as I think you were suggesting. So you can, if you do think, if your liver function tests are elevated on a blood test, having an ultrasound done of your liver to see if there's a fair amount of fat in there is a good move. If there is, cutting out sugar and saturated fats would be really a great move, or at least, reducing those significantly. I've had incredible results with people just cutting out their sugar. My whole thing where I wrote my first book occurred because I had one of the people in my lab, his son developed fatty liver and was drinking a lot of soft drinks. We just cut out the soft drinks and the fatty liver resolved in like 2 or 3 months. It was fantastic. There is a thing called a FibroScan, at least in the US, which is a way to kind of look for fibrosis. And of course, the liver biopsy is the gold standard. It is, I do think, and I suspect you agree with me, Simon, that just an increase in fat in the liver isn't really such a concern. It's real when it's associated with inflammation, because it's the inflammation that transforms that fatty liver into NASH and then to cirrhosis. And it is a terrible disease to get cirrhosis from fatty liver, and it's a real problem. It's especially high in people with diabetes, and we believe it's because this fructose is being generated from the high glucose. But of course, I think the saturated fat is playing a very important role as well. So everything you say about that's right.
Is there a pretty high correlation there in terms of, would you see someone with a lot of liver fat that doesn't have much inflammation, or do you typically see as liver fat's increasing, we're seeing simultaneously inflammation increasing as well?
They can definitely be distinct. You can have fatty liver without inflammation. And interestingly, if you just give fructose alone, you see some inflammation, but not a lot. And I think we saw the same with saturated fat alone in our mice. It's really that combination of fat and sugar that really seems to lead to the inflammation. It's conceivable that really high amounts of fructose long-term can do it, and high saturated fat long-term might cause cirrhosis, but it seems that it likes the combination. There's something about that combination that really stimulates the inflammation and fibrosis. Yeah.
Have you looked much into the synthesis of ceramides from saturated fats, and how does that play into all this story? Because we haven't touched on that, and I know we're getting right to the end here, but someone might be listening and thinking, why haven't they spoken about ceramides?
So fructose tends to block fatty acid oxidation, and stimulate lipogenesis. So it's actively increasing fat and in the liver by increasing synthesis and blocking the burning of fatty acids and stuff. In contrast, saturated fats seem to be increasing ceramides in the liver, which is also associated with driving fat content. I'm not absolutely certain how the ceramides are working. I think you might be able to explain that better.
No, it's new territory for me. I've seen it written about in a few reviews, but it's kind of that, along with increased lipolysis, seems to be the mechanisms that people suggest saturated fat increases liver fat through.
Exactly. So there's more fatty acids that are then taken up, and there's a stimulation of ceramides that apparently are important in the fat, in increasing the fat content. So they are slightly different mechanisms, and I think more needs to be done on this.
Right. Okay. Well, this has been extremely interesting. Thank you so much for coming back on and having this discussion with me. And I know you didn't have to, and we've exchanged so many emails. So I really appreciate your time and really just your passion for this space, and talking about the mechanisms and talking about the human health outcome studies, and just helping us make more sense of everything, and hopefully be able to make better decisions with our food choices. Is there anything, Richard, that you feel like you wanted to discuss today that perhaps we missed, or you wanted to expand on?
No, I think we hit the main topics, but I do want to compliment you. You are a total sleuth, and you investigate the literature, you find all these studies, and you're absolutely spot on with your syntheses. So I'm very delighted to be on this show. I learned a lot from you as well. And the best way to move science forward is having discussions like this that raise these new questions. Why is it that this happens when we see this in the human, and in this particular study, and in this study we don't? It really it gets me thinking about what I want to do next. So thank you.
No, and thank you. I think your openness, and I think everyone will hear this, and just ability, you have your hypothesis and your thesis, but being open to discuss science that might not be fully aligned but is explainable, and then thinking about future studies, it's a breath of fresh air. So I really appreciate everything that you're doing. And I know that you're going to be involved in lots of science going forward. So open invite on this show. Come back and sit down with us at any stage and share anything that you discover along the way.
Yeah. And I have one final comment, and that is that my daughter is a fan of you and thinks that you look like Johnny Depp. So
you did mention Jack Sparrow before, right?
Yeah, I did. That's came from her, I think.
Okay, we'll say hello to her.
Okay. Yeah, thanks, I will. Thanks, Tracy. Okay, thank you.
Thank you for joining me for this episode and your interest in science-based conversation. I hope you enjoyed it and found the information covered interesting and instructive. If you did and you'd like to show your support for the show, please subscribe to our YouTube channel, where you can stay up to date with new episodes and watch them in video format. Yes, the full-length videos. Please also consider subscribing to the show on the Spotify and/or Apple Podcast app, wherever you enjoy listening to podcasts. You can also leave a review on Apple or Spotify. Again, a great way to support the show and make our content more discoverable for others to enjoy and learn from. If you have any comments about the episodes, suggestions for future episodes, including guests you'd like to see on the show, or questions that you'd like to have answered, please leave those in the comments section on YouTube. I myself and my team will take note of these comments when planning future episodes. Finally, the best way to support the show and receive discounts on products we love is by checking out our sponsors at theproof.com/friends. Enjoy your week, stay well, and I look forward to catching you in the next episode.